All right, and I'm going to pull up the chat window too, because I'm going to have y'all working a little bit while we're in here. So welcome. I've got to tell you, my experience with teaching kidney, I think, is something that a lot of us can identify with. So when I was in grad school, I can remember my advisor told me that students would always say, well, I did really well on the kidney exam. Can I just, throw out a different grade? And he said, the problem with the kidney exam is it's the easy one because I don't even understand kidney. So fast forward years later, I'm applying for jobs, and what does everyone ask me to teach in the teaching part of the interview? Kidney. So teaching about kidneys it can be really difficult. What I want to do is I want to take a second here, and you'll have to excuse my eye in the sky. I want to find out what are the big learning outcomes that you are trying to reach for when you are teaching your students about kidney. We're kind of focused more on like urine formation and nephrons today. Feel free to pop it in the chat or hand up in chat. Urine formation, yes, nephron structure, that's a great one. Like, what are the parts? We'll come back to this list later, okay? Urine formation is huge. Yes, glomerulus pressure. And the countercurrent multiplier, yeah. So I teach comparative, and that's a big one in comparative because it comes up at a couple spots. Yep, structure and function. Oh my goodness, speak into my heart. Filtration reabsorption. Sorry about my chicken scratch. Blood pressure. Yep. All right. Renin-angiotensin system. Oh man, the students love that one. I love to kind of show it to them. And we have the hormones, ADH. Oh, you guys have all sorts of good ones. All right. Filtration rate, so GFR, and kidney disease. So we've got a nice list of things that we're looking at here, right? And I'm hoping to show you that we're going to hit a lot of these in what I'm going to show you today. And so just because you're using visible body doesn't mean that you're going to have to change your learning outcomes. It just helps if we go from the learning outcomes and build a lesson, we're going to build a better lesson than if we try to follow, whatever book, which is sometimes just the easy thing to do, right? Okay, so as I mentioned before, teaching renal physiology is really hard. In fact, I actually found this is an academic paper about difficult things to teach. And I want you to think about how many of them are on our list, right? GFR, homeostasis, urine concentration. Oh my goodness. So all of our greatest hits are apparently really hard to teach. And this is a paper from like a while ago. In fact, renal physiology can be so hard to teach, it can actually wind your teaching straight up in the toilet. This is a picture of a video I did a couple years ago, and I think it shows the desperation I was feeling with teaching renal physiology. So when we approach learning, the sort of stereotypical approach is this idea of students as a passive learner. The student is a sponge, they're soaking up information, and when we go to do assessment, we're able to just kind of get that same information out of them. But this sort of approach, it doesn't have the same lasting power. Once the sponge is dry, right, it's not going to soak up the same water again. So this isn't going to stick with the students in any particular way. And that's why we like to move to more so active approaches. And if you are a person who teaches in an active manner, I want to I'm hoping that I'm going to challenge a little bit your ideas of active and expand them. So when we think about a lesson, we think about a structure. Normally we have some sort of pre-work where the students will read or watch a video. Then they're going to come to class and learn. Now, if you're like me, when I teach with this structure, you usually hope that they will do the pre-work and they don't, but then they do it as post-work. And then you're going to have some kind of student output, which would be like their homework, their exam, their quiz. Okay, so let's take a look at that pre-work. Now, we want to think about how do we get students to engage with it? So how do we get students to, with active learning, we want them to engage with the material, think about it, sort of ask questions, answer them, instead of just passively absorb like that sponge. So we want to think about how is the student interacting with the material? Are they making connections? Are they reflecting? And I shamelessly grabbed this image from the internet. So it's not even about kidney, it's actually about Play-Doh. But we've all seen this textbook. Like we've seen the book where the student is just highlighting everything and they're not really filtering, they're not thinking, they're not really going through that active process of thinking about what is this thing that I'm reading and what questions do I have and how do I answer those questions. So I'm answering my own question that this is a very passive means of learning. When we look at the visible body materials, I'm going to give you an idea of what's in the toolbox. So there are a couple different things. We've got videos that are already there that are going to show how a process works. These are really good for kind of intros into process. We've got histology slides and 3D models. So the 3D models, the students can actually rotate around, they can dissect. There are buttons, and a lot of you are visible body users already, right? So you know the buttons, you know that they can click on it, they can read more, they can, and it's capturing that active curiosity while they're thinking about it. They don't have to wait until whatever page it comes up at. They can click on the thing and learn about it right then and there. And I think that's part of the power of it. All right, in addition to that, there are pathologies. And this has become an ever increasing list. This is, to me, the most disgusting one, the polycystic kidney. It looks so nasty. And these similarly are dissectable 3D models where the students can actually read more and learn more about what's going on. And there are modules where these things are packaged together in tours where you can have multiple images that are going through the material in different ways. So you'd have your where the kidney is in the body, your cross section of the kidney, and your nephron all in the same tour. And that's another way for students to access this information. And there's even more than that. So if I were to create a lesson, again, based on our sort of lesson plan that we created here, in my mind, I'd start with the videos filtration and absorption secretion, which just go through the basics of what these processes are. Have a nephron tour. Where's the nephron? What are the parts of the nephron? A little dissectable action. And then I love to wrap it all up with pathologies, which kind of looking at the learning outcomes y'all had. It seems like you're kind of in that same vein. So I would pick something like acute tubular necrosis and then say, okay, now that you know all this stuff, how is that going to affect the patient? So your sample reading, instead of being a textbook that's going to get way over highlighted, is going to be like, watch this video, read through this tour and dissect it, maybe find these structures and explain this pathology. And that would be sort of what you could set up in if you're a VB Suite user, you could set it up just like this. And your student, instead of reading and highlighting, is now really getting actively engaged and they're capturing their active curiosities. All right, I put videos in there because I was being a little extra when I made this. Okay. When we look at courseware, your sample reading in courseware can be very similar. Now with courseware, what you can do is you can put in other items. So I'll show you in a minute how to get to a course that I made. And mine always have guided notes. These are questions I want the students to think about and answer for themselves. And my goal is that they will come to class with those questions. So they don't have to come up with them all on their own on the spot. They can be like, hey, you had this thing in the guided notes and I have no idea what you're talking about. And it facilitates that. As Courtney said, I tend to work with really early learners and non-traditional students. So sometimes it's just really teaching them that process of studenting and kind of modeling what it looks like. Then you could have your modules. And the great thing about courseware is we can even put in labs, and there are existing labs, or you can put in your own document. It's all good. And you can put in quizzes. These modules are pre-built. They're also editable. You can download the pre-made course online. Shameless plug. My personal favorite is the Cindy Harley Anatomy and Physiology 2 right there. It's got a lot of those materials that feel free to use them. Any of the documents in there are Word documents that you can personally edit and make work for your course, because teaching with someone else's materials isn't great. It doesn't always match with what you want to do. Okay. And now we're going to get into immersive labs. So immersive is cool. This is like the new hotness in the visible body world. And one of the great things is you can put these modules together just kind of clicking and dragging, but there's an already existing course in immersive. And in this already existing course, if you preview the assignments, what it's going to have is text on the left. And a lot of times that text on the left is going to walk the student through what they should be looking at, what they should be finding. So now if you are an Atlas user and you've had students that don't really know how to interact with Atlas, now they have that, right? They have this, hey, rotate it this way, look at this. Here are the greatest hits of what I want you to see. And here are these high quality visuals. The text is minimalistic. And we really wrote it kind of aiming at the idea that this would be helping sort of those C students. If you've got your students that are just all stars, you might be supplementing with a couple more things. And again, you get all that functionality of dissection, being able to move things around, being able to click all the buttons and try the widgets. And it's pretty fantastic. One more video for y'all, because if you're like me, you're geeking out about this. So the best thing about this product is it is editable. You can edit the text. You can make it say what you want it to say. And so now we're getting a place where we can tell the students how to interact with the model. We can be kind of over their shoulder. And if doing that for all these models seems daunting, there's already a package that you can start from, and then you can put it how you want to. In addition to this, you can link out to other items. So I tend to use a lot of different simulations. And what I'm able to do here is I'm able to link out to the PhET simulations from University of Colorado, or able to link to a OpenStax chapter, or even to a shared Google Doc that the students will work on together. And it's all within that same course shell. So the students don't have to leave it and feel like they're dealing with multiple different things, which sometimes can be daunting from the student end. All right. So I want to take a second, and I said I was going to challenge your notion. So do you feel like this is something that has made the students potentially interact with the material in a different way than they would in a textbook? Do you think that they're engaging in that active thought process of taking control of their learning? Yeah, I'm seeing it definitely, right? So this is what I think is really exciting. Little spoiler alert, I just submitted an article, and it's about using visible body in teaching. We looked at 5 different institutions, and we found at all 5, we got an increase in the average student grades, and that we got a massive decrease in D's, F's, and W's, so students not receiving credits. And those didn't just go to C's, we ended up with an increase in A's. So we found that the students do better. I think the reason is that it's this active form of reading. And when I think of the modern textbook, this is where we've dropped the ball as academics, right? You move to a digital textbook, and your digital textbook is a PDF of a hard copy textbook. Not good. This is the next level. This is like, now we've got text. The instructor can modify that text. You can choose what you show, what you don't show, and you can have those models that the students are interacting with. Okay. So, oh yeah, and I'm seeing the chat right now. Yeah, many instructors and myself included, you know, there are times where you just kind of tune out and you're like working on a different course and then you come back and there's something new available. There is so much in visible body. And one of the things that I always have in my courses, and if you ever want me to share this, just ask, or I know that the folks at Visible Body have similar things. I always start with a how to use visible body because even the students don't know. And that way they can acquaint themselves. And what I do is I kind of, it's like a worksheet, you know, what does this button do? And it just helps them learn how to do it. And I tell them, you keep that worksheet, that's now your cheat sheet for when you forget how to do things. But I embed it pretty deeply in my course. Okay. So we're going to move to some in-class activities. So this is going to be coming into more traditionally what you think of when you think of your active learning. So we can think of, how do we have students engage with material in class? I could just talk at them, but that's going to get really boring, and it's going to get boring for the students. It's going to get boring for me. In fact, when I used to do it, I just felt like I was faking. And this is much, much better. So one of the things that I want to impress upon you is you can move away from PowerPoint with this product. One of the things that I do is I'll create a flashcard deck, which is basically I save views. And what I'll do is I'll present that deck to the students. So I can rotate a view around, I can make notes on it, and then I can move to the next card, which could be a completely different view. I can then share my annotated note cards with my students, or even better, I can give them the blank ones and they can annotate the note cards during class. This is a really fun way to kind of use what I think started as like a flashcard function, but has so much more power than just flashcards. And I'm a really slow typist. I actually got into playing with the pen function and drawing later in this video, and kind of thinking about, well, how would I explain podocytes and what would I be saying to the students? You could even assess those note cards and have them answer specific questions. We'll get into that later. So other in-class activities, and these don't have to use visible body, they're certainly better with. So I'm a big fan of card sorting tasks. I will go ahead and I will print out little pieces of paper with structure names on them, and I'll just put piles on the table, sometimes with some red herrings in there, and I'll ask the students to arrange the cards in terms of, in this case, it would be like where the structures are in urine flow. You have them do it in groups. They're going to be explaining it to each other. And you can tell them, hey, it's open visible body. So instead of open book, it's open visible body. Now you can use this and figure out where things are. Another thing that I do, and you could match the cards with anatomy, match them to function, pin it to the model. You can get creative here and just have some fun with it. Another thing that I do is I love to have my students draw. I think the more you draw, the more you know. So I'm really fortunate. My classroom has whiteboard walls. And so I'll just kind of throw dry erase markers at the student and I'll say diagram something. In this case, we would want them to do urine flow. You'll have to excuse my picture here. They were actually doing circulatory from the, like basically within the head. And then what I'll do is I'll have these groups kind of switch place and correct each other's work. And they'll have discussions about, you know, why did you do this instead of this, and who's right and who's wrong. So they're basically teaching each other. These are ways that you can talk less, which is actually if you're like me, kind of hard to do, but also really good. It helps the students take some ownership. And again, we do that open visible body. Now, you can also play games. So if you're a person who's interested in games, there is mostly it's free, but if you want the extra bells and whistles, you got to pay. That's called Kahoot. And Kahoot is like Jeopardy. So I've got a couple Kahoot examples: pin the tail on the kidney, place your pin on the kidney. The students would actually be working on their smartphones to put the pin where they want. Now, this allows you during class, actually all of these activities allow you during class to assess, do my students understand, do they know what they're supposed to know? Or are there big misconceptions? So I can do a little Kahoot example here. So what would happen in this case is the student would join. I have them start with ready-made nicknames. And then your questions are going to come up. You can have them do this asynchronously, by the way, if you're an online instructor. They don't have to do it in class, but it'll give them the question. And it's even got music that kind of stresses you out. And if we do it wrong, all right, let's do it wrong. Oh, now I know I'm wrong as a student, right? And it's going to give you their scores at the end. So as an instructor, this is something that you can hold on to. So doesn't that feel good when you get something right? It's like, oh, I did it. And you can set these up pretty easily within Kahoot. Uh-oh, what did I do? Technical difficulty. Hold on a second. I think Kahoot locked me out. Oh. Don't try to give a presentation with it. It will hurt you. Give me 1 second. There we go. Oh, that was scary. Oh dear. Technical difficulties. Don't worry, folks. We'll get back there. What's going on?
I can just add while you're looking for that, Cindy, that I did see the comment by an attendee that many instructors feel like they're not taking advantage of a lot that Visible Body has to offer. And that's common. You've got a lot on your plate. So we offer that the service of helping you take advantage of all that Visible Body has to offer. I believe Courtney posted something in the chat. We've got a support site with articles. We have a team of education team members, customer engagement specialists who will train you and help you build materials. They'll actually set up your course for you. So definitely take advantage of that. We're here to help.
Excellent. All right. And the supports are really fantastic, and the pre-existing sort of courses are even really good too, which I feel like is a rarity when you're dealing with publisher materials, but there's a really fantastic. There's like a special flavor for everyone. So one of the next tasks I want to take you through is the idea of building a structure. So one of my colleagues teases me and he says, Cindy, you're showing your engineer, because I went to Case Western, which is a school that's more of an engineering school than biology, but hey, it's all good. So this is the first glomerulus I built for my students. I wanted to kind of give them this idea of glomerular filtration and explain how we can change glomerular filtration depending on like the constriction or dilation of those arterioles. So in this one, I have a bunch of lab tubing and I connect it to the sink and we look at how much comes out the glomerulus or the afferent arteriole, and I literally sit there and pinch it off. It works in a small group. It doesn't work in a large group. So I started thinking, well, how can I build the glomerulus that'll work a little bit better? And I went with the toilet paper tube first, so you don't have to get fancy. You can just raid the recycling. And that one worked. It was a little messy. And then we got to this one, which I will show you. So I have, let's see, a Gatorade bottle here. I kind of connected it. So I've got two tops and I've got a bunch of holes in my Gatorade bottle. Okay. Now I took the caps and I got fancy dancy and I made different diameter holes in those caps. Okay. So this is like straight up MacGyver stuff. And what we can do is we can actually, oh, it's going to get a little fancy. I may have out-fancied myself on this one, but y'all can imagine. And you can have the students come up with these ideas too, and you can basically tell them like, hey, build this thing. And if it doesn't work, it's fine. You just ask, how can you make it better? So one of the things we can do is I'm going to twist this, and I've got my various things in here that are going to represent components in the blood. So I'm going to have white blood cells and red blood cells and sodium and urea and whatever. And so what you can do is you can actually just put this over your bowl. My bowl is the tube. So what's getting into the tubular fluid, and the students very quickly are going to notice that what's getting into the tubular fluid is going to be, whoop, my sodium and, you know, water and urea or whatever. And they're going to notice the big stuff, the proteins, the white blood cells, the red blood cells, those aren't getting through. And then you say, well, why? And they'll say the holes. Oh, so what does a glomerulus do? Filtration, yeah. And then you can even ask the questions, well, how could we prevent more stuff from getting through here? And that's where you can get into the podocyte discussion, because the students will almost always say, well, you just cover the holes. And you can get into that. Now, I took this up a level. And admittedly, I just started playing around with this a bit ago, trying it to work perfectly. But one thing we can do too, ooh, it's a little bit messy. That's okay. I like making a mess. If you're not making a mess in science, what are you making? So I've been working with a very, very, very dilated efferent arteriole. Okay. But you can imagine, well, what happens if we constrict that efferent arteriole? And now we can look at how does that affect that glomerular filtration? And what'll happen pretty quickly, and I'm trying not to make too much noise here, but you can imagine that this glomerulus is going to get really clogged up really quickly. That little stuff's going to come out, and we're going to get more out because there's back up in here and the stuff wants to get out. So this is kind of your MacGyver way to work on teaching glomerular filtration. There we go. And it's literally with things that I got out of my kitchen and out of my kitchen pantry and out of my recycling. And all you need to do is just kind of go through and say, oh, well, all right, here, this garbanzo bean, that's your red blood cell. This is your white blood cell. You know, maybe oats or platelets, I don't know. This is very similar. There's a pre-built kit from Carolina that works in a very similar way. I think that you learn a little bit more when you try to build your own stuff. And the reason that I think that is that you actually have to think about, well, what is working? How does it work? And how am I going to make it work? And so you could even put that on the students, especially if you're an asynchronous person. Hey, try to build something that would work like a glomerulus. Even if they don't get it perfect, give them the out that even if it's not perfect, they can tell you what they would do to make it better. And so like with this one, hey, how would you make it better? And they may say, you know, fill it with water. They may say, well, you know, I kept getting white blood cells out, so maybe we need to make those holes smaller. All right. So moving on, and this one is in your packet. Within the packet, there's this activity. It's going to look cumbersome at first. This one was come up with, or a student teacher I had came up with this one. She did a beautiful job. I'm going to show you how to make it work. And then the materials that you need are in your packet with a little explanation, because I know it looks weird, but it works really well. So we're getting now, we went from filtration, which was, again, glomerular filtration was one of our big learning outcomes. Now we're going into that loop of Henle, that countercurrent multiplier. We're going to start with the regular loop, and then we can get to countercurrent from here. So what you do is you have have a bunch of little cards around, you tape out a nephron on the floor, have a bunch of cards on the outside that represent the external environment. And then usually what I do is I have a basket and I put my cards that represent the internal tubular fluid in that basket. And what I'll do is I'll walk to 300 milliosmoles and I'll look at the students in there and I'll be like, okay, how are we going to balance this? And they'll be like, what? And you'll say, well, we're in the descending limb, right? What is that permeable to? Oh, it's permeable to water. Oh, well, what would balanced water look like? In this model, we're taking an oversimplification. We're literally balancing it numerically. So if we were to count the waters up, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 on the outside. I believe that we have something like 13 on the inside. So we say, okay, well, we're going to move 2 over. Now our water is balanced inside to outside until we move down to 600. And now we move down to 600. We're going to move some water to balance the water. And so we do that. And again, it'll tell you on the handout how many move and also what the concentration is going to be on each side once they do move. And we keep stepping through the loop of Henle this way. So what this helps the students with is you're going to have students, there we go, now I get to the bottom, we're going to go up. Ascending limb, what's ascending permeable to? Hopefully they'll say sodium. If they don't, it's okay. And here we balance using the number of solutes, okay? So we've got 2 urea, and I've already moved my one of my sodium. So we've got 2 urea and 5 sodium. So 7 solutes on one side. And now what we're doing is moving those 7 so that we'll have even number on both sides. We've got 5 over here. So we'll make it so it's 6 and 6. Move 1 over. Boom. And it can only be sodium because it's permeable to sodium. So what this activity helps with is it helps the students internalize that you're permeable to water in one part, you're permeable to sodium in another. It helps them to think about that concentration gradient and why it matters. And what I love to do at the very end is say, what have we accomplished? Because I remember when I was a student, I was like, okay, you removed water and you removed salt. So like, what's the point? Well, the point is, right, that now we've concentrated the waste. And the cool thing about this activity is you can actually expand it. So you can make the loop longer. So I do comparative physiology. I love making the loop longer or shorter and saying, well, what does the concentration look like? Where would this animal live? You can also try to make it more accurate, play with the osmolarities. You can add glucose to it. And literally when I add glucose, I take those cards and I'll throw the glucose cards into my basket. And what will happen as the students are kind of moving through this tube is it's harder for them to find the waters. And so there's going to be less water reabsorbed. It's not an ideal, super accurate situation, but because you end up with more water coming through as a result of solutes, or you could tell them each glucose needs to hold on to at least a water, then you're going to show them, hey, what is glucose doing there in concentration? And hopefully they'll realize that it's getting more, that there's more water being held on to. You can also have, when you're done, now your students are going to look at the outside environment and they're going to be like, but this doesn't make any sense. And that's where you create a second loop and you have students working as the vis erecta. And what they're going to be doing is collecting those waters and salts in the appropriate places. And that's going to give them an idea of the vis erecta kind of maintaining the concentration gradient. If you want to get real fancy dancy, you can have them doing it at the same time, but I would do that after you do the first one. And then your students are going to be able to see that counter current multiplier in action. All right. So we're doing pretty well with our active learning activities. When I look at our list, right, we've gotten through glomerular filtration, the pressure, the counter current multiplier. So we're doing pretty well on the list. As we go through here, now we're going to look at how are we assessing students and can we make that assessment more active? All right, so now we're looking at the output. All right. So your traditional output could be a handout, and these exist. So no shade, we are busy, busy humans. There are some fantastic already existing handouts, and if you use courseware, there are even some that are already like auto-graded handouts that you can use for your students. You can put quizzes in any of these programs or do them in the classroom. You can also do, your exam or lab practical, which you probably already do. But let's think in a different way. Let's try to think about being more active. One of the things that I love to do is group quizzes. So I print out my quiz, same questions, both sides. I'll have the students take it individually, and then we flip it over, we get into groups, and they ask their groups questions. This allows them to correct each other right then and there. So what the students are doing is you're avoiding the, I got my grade, now I'm going to stick this in my bag and like never pay attention to it again. They're actually thinking about it and they're going to retain it better. You can turn the group quiz into a game, right? You can put them in teams and do the Kahoot or some other kind of game. You can have the student lead the lesson. So now you're going to be quiet. You're going to give the student some kind of, here are the things you have to explain. Have them lead it. You can have a discussion about pathology or give group scenarios. One of the ways that this manifests in my class is that we have what we call science communications. I'll admit the one that I'm showing here as an example is not my best example, but it was about urinary. And so I wanted to use it. So what my students will do is they will create some kind of communication that talks about what the condition is, what is the anatomy and physiology affected by it, what are the treatments, and they will explain it in a way that's accurate. You could have your students use this to teach material, or even do a gallery walk, have them put them up around the room, and now they're going to give each other feedback. All right. I like to get a little extra crazy. So one of the things I started doing a couple years ago, and I think I started this around COVID. Morale was low in the class. The students needed something new to do. So we started doing memes. And the great thing about memes is that they're funny. And so my students are on Slack, and the role was you've got to create this meme. It's got to be correct. We're going to judge it on originality. It's got to apply to stuff. You get an extra point for creativity, and you have to give me a 2-sentence explanation of what it shows. And the students would do this. They put them on Slack, and we even had a thing where, depending how many thumbs ups they got, they would get an extra point. Super fun. You could even have it so other students have to explain the memes, and that's always a good one too. And it's a way to get sort of your students interacting with each other, especially if you're in an online asynchronous environment and you're struggling to get that interactivity. It's a good way to do it. It's also something they're very familiar with. All right. I also do sketch notes. So rather than exams, I've moved to a system, and I gave a talk about this, so I'm not going to go through the whole spiel again. But the idea is that the students are going to be presenting the material in a visual format. They have to think about what to present, how to present it, what concepts are going to come across, and then they have to explain those concepts. So I actually argue most of my students say it's harder than an exam, but it sticks. And you can allow them to use visible body to create the visuals. And that takes out that, but I'm not an artist bunch. And it allows them to use the app. And again, you're reinforcing that app usage. Okay, there we go. Another idea I alluded to before is playing with the flashcard function. So here we've got flashcards that already exist, but maybe what you can do is put on a prompt, label the structures and list their functions. And your students could type out their answer and send it back. Or if you want to just know if they're getting it, right? There's the summative assessment where we're getting grades, and then there's the formative where we're just kind of finding out what do they know? Are they with us on this learning journey? You could have them write down areas where they're confused and send those to you as a muddiest point card. So I don't understand this thing here. And then you know what you need to touch on in the next lesson. Okay. Last but not least, one thing we do in my class, and this has been one of the biggest changes we made, it's one of my favorites. So I've got the sketch notes, I use those predominantly for physiology content, and then my students do video walkthroughs. They're very used to interaction videos, TikToks, that sort of thing. And so for the students to do a video walkthrough is not really a big ask. In fact, they don't really need my tech know-how, which is great. So here's an example.
So in the kidneys, what's happening is, first of all, the main function of the kidneys is to take the blood and filter it, and then all of the gunk that filters out, there's some reabsorption that occurs and secretion occurs.
And so I'm skipping ahead.
So the Bowman's capsule, the round part here, collects all the filtrate, which then enters the proximal right here, kind of whitish.
And so here you're getting an idea. That student can explain that anatomy. They know the anatomy. And it's not a big deal if they have to do this like multiple times to get through it, because that's how they're learning. And so now you've taken their assessment from something that they're just doing for you to grade, and you've turned it into its own unique learning opportunity. And this has been super fun. If you do it, I would recommend setting a time limit, because some students will take like 20 minutes and you don't want to spend the rest of your natural life working on that, because it is a bit much. So, whoops. Where did I go? Oh dear. There we go. This is why, I mean, it's better if my students don't watch my tech know-how. So hopefully we've gotten little pieces of each of these learning outcomes, right? We didn't get into the hormones. There are visible body assets about those, and that go through those, but we got through the countercurrent multiplier, we got through glomerular filtration, a little bit of urine formation, and that structure and function, and there's even more there. I'm just touching on the tip of the iceberg. So with that, and I apologize for talking so long, I usually don't. I would love to hear maybe ideas, thoughts you all have, and we can just figure out how we're going to make this a more amazing year for teaching kidneys than last year was. So I do see a little note in the chat saying, do I share slides with my 3D simulations? Yeah, you can share the note cards, and then the students can take notes. I used to do just lecture from visible body, and my students had a hard time taking notes. They found that they were drawing a lot. And so one thing you can do, and this is how I started, is I think before the flashcard function, I would make like pieces of paper where I'd paste the images on there so that they could annotate those pieces of paper, because I knew the order of the images. But I think now, if you use the flashcard function, they'll be able to take notes right on those flashcards. It's pretty powerful.
And I'll just add that the flashcards are both web and mobile. So if they take notes on their computer in the flashcards, those notes will show up in their mobile app flashcards as well. And they don't have to do anything special. It syncs behind the scenes. So it's a pretty cool functionality.
And one of the neat things you can do too So I'm a user of Notability. I do a ton of stuff in Notability. I'm a big iPad user. You can literally just take screenshots of the visible body models and then annotate them in Notability. So you can move them to, or Good Notes or whatever program you use. If you're an iPad user, that's a really good way to do things. So I love this question. Do you think handwritten notes on the figures or flashcards are better? I like handwritten. And I like handwritten because I'm convinced that since you kind of need to go through, visual cortex to motor cortex to frontal, right? I think because you're using more of your brain, you're actually going to retain more. So I actually do try to get my students drawing a lot and interacting with the material. If you don't, one of the things that you can do if they're taking digital notes is at the end, have them sit down and write down, the three big topics. Or what I'll do with my non-majors is I'll seed them with words. So write me 5 sentences about the kidney and you have to include these words. And the students will actually think about, well, how am I going to use those words? And it allows them to reflect on the lesson. It's a cute little trick that they don't find too intrusive that allows them to retain that information. All right. So what I would say is, there's a question about a visible body scholarship. What I found with my students, so I work with predominantly with low-income students. I work in a small department with a low budget, it's even lower these years. This software is actually going to be cheaper for you to use than the 3D models, which can be very expensive. There are a couple ways you can do it. One way is if your university does have the budget, you can have it as a classroom license, and then they can use it when they come into class. I don't like that quite as much because then they can only use it in class. My goal with the students is to be on their phone, because if I'm on their phone, now they're like my captive audience. So I have my students buy it instead of a textbook. And it works out that it's about $50. Am I remembering the price right, Emily?
Yes.
And that license is good for a couple years. So I worked with, at the time I was teaching A&P 1 and 2, but I actually worked with our instructors of our nursing program and said, can you use this too? And now our students don't mind because they use it in so many classes and they get to kind of keep the software. So that works really well. I don't know if there are any options for low income subsidies through you guys.
There are sales sometimes, but not for courseware. But we have kept the prices low. We've never raised the price of courseware since it came out in 2017. So that would be, we do try to keep it low, lower than any textbook or any other software out there for assessment. The other option would be, you can do a free trial. It's just limited. It wouldn't get you for the full year or the full semester.
Yeah, you might, so I'm seeing something included with tuition. You might be able to do a lot of programs with labs, right? Have that differential tuition, and it might be something that your department or program lets you use the differential tuition for. And even if, so let's pretend that, your students can't afford it, university can't afford it. If you can get a license, back in the bad old days when I started using it, I had the license and I was telling students that were really struggling to buy it because courseware didn't exist yet, but I would still use it with my teaching and I would project it and use it instead of lecturing and be like, okay, we're going to look at the skull now. And I would rotate it around and take those bones out. And the students loved it because they felt like I was talking to them more and it wasn't death by PowerPoint. Instead, it was more of a conversation about structures and what they looked like. And I even would use the videos sometimes to like get a drink of water. So there are definitely ways to play with it. Now I've got a big honkin' touch screen, and I use that sometimes for lecturing, and it's super fun. The students love it. All right, I know I hit you guys with a lot of ideas, but the idea of this isn't that you're not going to do all these things, right? Because that would be, it's a lot. But if you just think about it and think about different ways to do things and change one thing that you're doing to be a little more active, or maybe change how you're using visible body and try one new piece, that's okay. And if it's an assignment, you can always start it as like an extra credit thing just to see how it works.
All right, we have about 4 minutes left. Does anybody else have any questions for Cindy or for us?
Yes, I know some of you are probably, I don't know, I feel like a lot of us have been adjusting to this more so somewhat hybrid space that teaching is now. And one of the things I love is even if your students won't do the reading, they'll watch the visible body videos. So you can be like, hey, watch these 2 videos before class and then come in, and now they've already got that grounding. And the videos are short and they're good. And so they'll go through them. Yeah, the countercurrent multiplier activity is fun. I would recommend like do it on your tabletop once so that you're comfortable with it. It works really well, but when you look at it first, you're like, oh. Oh yeah, Visible Body has great YouTube videos, and they keep coming out with more and more.
It's so validating to hear. I'm glad that you're using those videos, Margaret. That's awesome. We love making them, so very glad that you're using them.
It's always fun to know they're getting used too.
All right, so I guess we're gonna probably wrap up. We have about 2 minutes left. So I wanted to thank everybody once again for joining us. And thank you so much, Cindy, for your fabulousness and all of your wonderful ideas. And everybody keep an eye out for tomorrow's email. Like I said, you're gonna be getting a kit with these activities and these rubrics and some other supplementary materials. We're going to throw in some of the labs that Cindy had touched upon, our visible body labs, and lesson plans, some flashcards, all that goodness, as well as the recording from today. So keep an eye out for that. Sorry.
You've got a question in the chat of it, can they share it with other people?
Yeah, I mean, I don't see any reason why not. I mean, Cindy, these are your activities and everything. I will say for, the labs and the flashcards and the tours and everything, you will need a visible body and courseware instructor account. If you don't have one, I'm going to put in the email a way that you can get in touch with our education team to get one. So, so keep an eye out for that. And yeah, I'll be throwing in some other follow-up goodies, including the amazing series that Dr. Harley has been working on with us, the Visible Biology series, where she goes through some tough topics in biology using visible body, and they're very funny and they're very fun, and I highly recommend everybody watch those videos.
And if you have any questions or you run into roadblocks, just reach on out. And I will say, before I started using visible body, I was kind of the burned out, the poster child for burned out A&P instructors. And I started using it, and it just has changed the way I teach. And every year I find something new to do, and it's made it better, but also just for me more fun and invigorating, and the type of thing where I will make a glomerulus out of a couple Gatorade bottles because why not? So it's really changed me, and I hope that you have a similar experience.